All right. Hello, everyone. Welcome back to the SNRE Graduate Seminar, and welcome to those of you joining us on YouTube today. We are going to have a presentation from Sergio Marconi, and Dr. Ethan White will introduce him. So go ahead, Ethan. All right. Hello, everyone. Welcome back to the SNRE Graduate Seminar, and welcome to those of you joining us on YouTube today. We are going to have a presentation from Sergio Marconi, and Dr. Ethan White will introduce him. So go ahead, Ethan. Right. Hello, everyone. Welcome back to the SNRE Graduate Seminar, and welcome to those of you joining us on YouTube today. We are going to have a presentation from and Dr. Ethan White will introduce him.
Yeah, we're all hearing that too.
Hello, everyone. Welcome back to the SNRE Graduate Seminar. Oh my gosh. If you have the YouTube page open, it's on delay. So that may be why.
Oh, that was me. Sorry.
All right. Well, it's my sincere pleasure to introduce Sergio, who's been a PhD student in my research group for the last 5 years or so. And while I've never told Sergio this before, expectations were quite high for Sergio starting early in his graduate career. When interviewing students to join us for graduate work, a key component is the perspective of our current lab members, though they're often a bit hesitant to pass judgment on prospective students. And I tell you this so that you can appreciate what it meant when, at the conclusion of Sergio's interview, one of my best and most understated students walked into my office and said, "I don't know what to say about Sergio, except that he is some kind of a genius." And so when I say our expectations for Sergio were set high, I mean they were set unreasonably high for him before he even joined the lab. So has Sergio lived up to these lofty expectations? Well, he's compiled a tremendous body of interdisciplinary scientific work over the past 5 years, requiring expertise from botany to machine learning to high-performance computing. And Sergio brought a broad and integrated approach to this work, not only writing papers, lots of them, but also developing software, publishing data sets, and running data science competitions. And he accomplished all of this in a way that made him a joy to interact with. Our regular meetings were always a highlight of my week. Sergio would bring in a stack of new results, and we would spend the next hour talking as colleagues about next steps and how to solve challenging problems. He was always so excited about the science that he could regularly be heard from my office explaining what I came to think of as Sergio's catchphrase, "I love it." In addition to his own work, Sergio was also always excited to help others. Anyone asking Sergio for help on something would get a "yes, that sounds amazing," or another "I love it." He collaborated with others as both the domain specialist with computer scientists and the computational expert with biologists. He organized conferences and data science competitions to bring together researchers from across the globe. And he helped other students learn advanced computing by organizing and teaching workshops. And Sergio isn't just a great scientist. He's also a well-rounded person. He paints, he cooks incredible Italian cuisine, he's got an adorable dog that may or may not make an appearance during today's seminar, and is just a generally amazing person to be around. So in summary, not only did Sergio meet the high expectations that were set by being described as a genius before he even started, he exceeded them in spectacular ways, because not only is Sergio an exceptional scientist, more importantly, he's an exceptional person. And so with that, I will apologize for once again setting the expectations for Sergio unreasonably high and let him tell us about some of the exciting work he's done over the last 5 years.
Wow, thank you. that was much more than I deserve, but thank you so much for your kind introduction, and thank you all for being here today. Before starting, I would like to acknowledge that today's talk is being held in the unceded territory of the Seminoles and the Timucua tribe, The latter, unfortunately, extinct mostly because of European colonialism. I think it is important to acknowledge this, to have us all reflect 1 second or two on the responsibilities that we have and take action against the oppression that continues today on Native American communities, including food insecurity and reduced access to higher education, among others. Ecology is hard, not only because its ultimate goal is to understand the fundamental rules that tie up the organic and the inorganic world together, but also because these processes are often influenced by the scale at which we study an ecological system, along with the processes that intertwine across multiple scales. For example, microclimate drives whether a seed can grow into a young plant, but plant-to-plant interactions have a major role in how a forest patch grows. But ultimately, land management is the dominant driver that defines how the landscape looks like. And more complicated about this is that all of these processes work together. Anyway, regardless of the scale of interest, field observations are central to ecological studies. To estimate the amount of timber produced by a forest land, we go in the field, we measure the size of a sample of trees, and we apply some models to scale those observations to the entire forest. Likewise, to estimate the role of plant photosynthesis and respiration on the global carbon cycle, we collect data of leaf chemical properties, and we measure the amount of carbon exchange, but just for a handful number of sites. And similarly, we identify tree species for many individual plants sampled in few dozen plots to measure the biodiversity and ultimately understand how species change the distribution across time and space for large scales. However, one key challenge common to all these examples is the inevitable sparsity of field observations. Collecting data from the field is time and cost consuming, especially considering the lifetime of a project. It's often no more than a couple of years. We often need to accept the compromise of having to do ecology with what we have, even if the sample size is often too little and potentially underrepresentative for making meaningful large-scale decisions. But what if this is not universally true? What if we can leverage the information held in imagery to link properties of field observations to objects that can be seen from the sky? This could potentially leave us with, yes, relatively less precise measures, but for potentially any plant in our study region. And in the last few years, we have been experiencing a rapidly growing number of satellite and airborne sensors built to collect geo-explicit information at a very high resolution. This favored the rise of remote sensing, this discipline of converting images from satellite or aircraft into meaningful information without making physical contact of the object, and often by using advanced machine learning applications. And trees and all this are extremely good examples of the huge potential of remote sensing. Trees are big objects that can be detected and delineated from very high resolution airborne imagery. They are rooted to their geolocation. It means they don't move, but this makes them even more easily detectable, not only through space, but also through time. Moreover, trees have been studied in remote sensing for quite a lot now, and they showed a strong signal both in the visible and the infrared light, which makes those objects not only detectable, but also characterizable. And what do I mean with characterizable? Well, I mean that we can measure some of the most important biological properties of the trees used to address large-scale or cross-scale fundamental questions. Today, I will talk to you about how we integrated remote sensing and field data to derive measures of tree size, species identity, and leaf chemical and physical properties, the traits, for individual trees and across a continuum of spatial scales. Leaf traits were the first dimension of plant diversity that we explored. Leaf size, mass, and chemical composition are fundamental properties that can influence core demographic parameters of individual plants, including survival and reproduction, for example. And these can vary more or less dramatically within and across species. Different species specialize to different environmental conditions, often by tuning those leaf traits, and they do so within a range of possible values. These ranges are generally constrained by what we call trade-offs, or that they can be found globally across pretty much all vascular plants. Now, these trade-offs are often referred to as the leaf economic spectrum. The leaf economic spectrum essentially represents the graph here on the right and describes a gradient of different plant physiological strategies, ranging from those who invest in durable leaves with lower photosynthetic efficiency, like the larch here, and those who invest in highly efficient but fast turnover leaves, like the aspen here. The leaf economic spectrum is a model based on strong limitations on soil nutrients, environmental conditions like climate or access to direct light. And despite these trade-offs being particularly evident across species, plants can adapt by plastically changing their strategy, also within species, which can result in rising of within species or even within individual leaf economic spectrum. This makes it even more complicated then, because the problem with understanding how leaf traits vary within species is that field data, especially traits field data, are often sparse, incomplete, and unbalanced, making it particularly hard to address the effect of ecological drivers on intra-species leaf economic spectrum. In this context, leveraging remote sensing to build approaches that can detect and label species, tree size, and leaf traits for millions of individual trees across a gradient of environment and ecological condition can unlock unprecedented opportunities to study how these biological properties scale within and across taxa. To make all this possible, we need to build pipelines. And in general, those pipelines are made up of three major steps. The first step, we need to develop methods for identifying individual objects, individual trees in this case. We want to do it algorithmically and from generally very high resolution images. And once we have the position and the dimension of trees, we want to align remote sensing data to those field observations. In this case, for example, we need to identify which pixels to assign to each specific tree for which we have traits measures. Once we have this match and the corresponding portion of the remote sensing image to the field, it is possible to build machine learning algorithms and infer the underlying relationship between properties of the image and properties of the object, which is, in this case, the tree. And then, after proper validating how precise those relationships are outside of the data used for training, we need to develop approaches to scale those predictions for entire regions of interest. Of course, all this requires the step 0. We need to have sites collecting all this data, and that's far from common. Fortunately, in the last few years, we experienced the rise of the National Ecological Observatory Network, which is essentially an holistic continental observatory that offers measures of both remote sensing and field data, and more importantly, also collected within the same time window. Airborne data from NEON consists of 3 main sensors: the first, RGB, hyperspectral data, and LIDAR data. RGB is essentially like the images you can take with your phone, with the main difference that each pixel here represents a specific GPS location on Earth. RGB data are often very high resolution, but collects information only in the visible portion of the light spectrum, which it's a bit sad part because it's so high resolution, but still it is not very representative of the properties of vegetation that instead are more clear in the infrared. In fact, on the other end, hyperspectral imaging represents pictures that instead of having the RGB channel has 426 channels. And each channel represents a 5 nanometer wide window of the spectrum between the visible and the infrared. And measures of reflectance recorded for these tiny bands allow for precisely identifying highly informative regions of vegetation properties, like, for example, the leaf traits. However, this comes with a price, which is the significantly lower resolution of the images. Finally, NEON also consists of LIDAR data, which differently from the other two, consists of not real pictures, but much more in actively shooting beams of light from the aircraft and recording their time of return on the sensor, which basically give us point cloud of estimates of height and structure of the vegetation in the scene. Together, these three data sources can offer extremely valuable information for identifying traits, tree size, and species. But yet, alone, they are often of little value without a way to convert their information into meaningful ecological properties. And even tree objects may be likely too hard to be identified by the human eye. And so we need a way to figure it out where the object is in an algorithmic way. For this reason, we need not only field data, which is necessary for machine learning, but we also need good estimates of how field objects align with image. Fortunately, for this project, we had access to some special data collected by the amazing Dr. Sarah Graves. This data was special because it coupled measures of leaf traits to precise estimates of tree crown polygons. And those estimates were drawn directly on the hyperspectral image from the field, which essentially nullifies the uncertainty in what is the pixel associated to a crown. At this point then, we have the data. The only thing that we really need to do is develop the steps of the pipeline. The first step, crown detection and delineation from the remote sensing images. The second step requires making estimations of tree size and of leaf traits. And for this project, the leaf traits estimations were for nitrogen, carbon, leaf mass per area, and phosphorus, which are four of the most commonly measured leaf traits all across the world. And finally, scaling predictions for dozens of thousands of hectares, which requires like scalable algorithms. For the first step, we use an approach published by Silva and collaborators in 2016 that proved very successful on the southeast forests. This method is based on using LIDAR to assign the highest pixel to the crown tops, and then applying a growing region algorithm to clump all the pixels surrounding crown tops and define the size and the shape of each tree algorithmically. Once we had estimates of tree height and crown area, we used this information to build allometric models and infer stem size. Allometric models are essentially regressions that try to couple some different portions of the plant using some underlying generalizable relationship. And to do so, then we linked few hundred ground measurements of stem diameter to the algorithmically derived crowns, and we overlapped them with the data, and we created this relationship between the height of the tree, the crown area, and what we were interested in getting, the stem diameter. To make estimates of individual tree traits instead, we have to develop a new approach that leverage differences in the properties of pixels that make up the crown. And we focused on providing reliable quantification of the uncertainty associated with those predictions. To do so, we use partial least square regression for reducing hundreds of those hyperspectral channels into just a few dozens of predictors. So the only thing that you may be interested in here is essentially a dimensionality reduction approach. And we use those derived predictors to build generalized linear models. And that's pretty much what it was. And by this approach, produced reliable estimates with squared R on held out data ranging between 0.5 and 0.75. And also, it produced good estimates of the uncertainty and was fairly robust for predictions on algorithmically delineated crowns, which means for all those crowns for which we didn't go in the field and get droning around the hyperspectral image. This allowed us to scale our approach for millions of individual trees in a space continuum that allowed us to identify with precision leaf traits distribution from a single tree to a plot, to a patch, to the entire landscape. And we could apply this pipeline to more than one site. And in fact, we applied it to 2 NEON sites. One is here in Gainesville, quite close to Gainesville. It's at the Ordway Swisher Biological Station. And the other one is the Talladega Forest, which is in Alabama. All these results, I want to tell you that all these results are actually publicly available and can be used for investigate how topography, management, microclimate can affect separately or altogether forest structure, like the distribution of tree size, or leaf strategies, like the trade-offs among traits, which is the leaf economic spectrum. And in fact, we use this data to explore how well our estimates match with the global leaf economic spectrum. On one hand, this allowed us to further validate that our models produce estimates that also make biological sense. But also, this allowed us to grasp some relatively new biological insights. For example, interestingly enough, our results matched with the global trends and ranges of the global leaf economic spectrum, despite the limited number of species and the relatively little geographic extent. This can suggest us that variation in the local environment may be driving a landscape scale leaf economic spectrum, which possibly can be driven by both intra and interspecies variability in leaf traits and leaf strategies, sorry. Moreover, making estimates for different sites allow us to compare traits distribution across a multitude of landscapes, with the potential to answer questions about how local driver may affect leaf strategies, also like different kind of forest or very similar kind of forest for a variety of forest types, for a variety of climate regions, for a variety of topographic and landscape structures. And in fact, this approach can essentially be extended to essentially any site on NEON, since they are gonna collect leaf traits and remote sensing all together. And that indeed is actually what we have been doing for the next project, deriving tree species class for all individual trees algorithmically detected on landscape for 23 different sites across the continent. For this project, we used NEON field survey as field data. And this data consists in vegetation, includes species class and stem size for individual trees. And each one of those individual trees is assigned to GPS location. From this data, we extracted all trees that we believe identifiable from space, which produce a data set of about 80 species, which, just to give you a hint, represents around 20% of the total amount of species that can be observed nationwide. Again, our pipeline consisted in the same main steps: crown detection and delineation, crown size estimate, development of most accurate machine learning classifiers as possible, and ultimately scale up those predictions for this time hundreds of thousands of hectares. For this project, we use a different crown detection algorithm developed this time by our amazing postdoc, Ben Weinstein. This approach allowed for identifying crowns directly from the RGB image, leveraging the fact that he has much higher resolution than the LIDAR. And this method is based on deep learning architecture. It's very easily scalable, is generalizable across the continent, and essentially performs twice as good as most of the other available approaches that are openly available. I mean, look at this box plot over here. Ben's algorithm is the purple ones, and all the others are all the other box plots. For the species classification, I want to spend a few seconds because we built a essentially more complicated-ish pipeline. This pipeline is built on two steps classifier that is based on first making predictions for 4 different machine learning approaches, and then use the vector of probabilities generated by each classifier as features for an overarching meta model, which we ultimately use to give each single box a species label and a confidence score, and each single box represents a single tree. This kind of approach is generally referred to as model ensemble or model stacking. The strategy is very common, and it gives the advantage of leveraging the strength of very different algorithms that may be better than the others just for particular sites or species or regions. And so if we allow for all those different methods to be intelligently integrated all together, this allows us to build models that are more generalizable and provide more robust predictions and associated estimates of the uncertainty. And once we also had species prediction, we could improve even further our allometric model. And again, the allometric model is the model that we use for scaling tree properties and infer the size of the stem. In this case, we leveraged our best guess of individual tree taxonomy, that is our species classification, for building a multi-level Bayesian model. Now, this approach allows to leverage the effects that all different species and the relationship among species can have on the parameters of this general relationship between height, crown area, and diameter. And actually, it produced very good estimates, with the bigger errors being often attributable to one single sparse species from one single site, and not even for all of them, but essentially for most of the crowns for which the crown area was actually very tiny. And so probably they were not aligned well with the actual ground point. Anyway, the model, the species classification model, provided fairly decent accuracy with an average of 76% of trees collectively identified on held-out data, again, on 23 sites that span from Alaska to Puerto Rico. And just to make clear, this is one single model that leverage also the site as an information, but this is a single one overarching model that tries to make prediction for 80 species, which is kind of like unique in remote sensing applications right now. Accuracy seems to be lower in the eastern of the United States. And in part, this is because of the presence of few fairly high plastic species and of many oak species that in general get misclassified among each other. But also, this is probably because eastern forests are more diverse and show a generally higher number of species. What you can see in this graph, for example, is the number of species at each site on the x-axis, while the classification accuracy on held-out data is on the y-axis. Now, with the increase of the number of species, our classifier clearly showed a reduction in the accuracy. However, it still outperformed our baseline model mostly all the time. Well, with the exception of one site that we probably don't suggest people to use for ecological application. Anyway, you may tell, all these sources of classification, don't they reduce the confidence in using these derived surveys? I mean, we're still talking about 20 to 25% of misclassification. That may be a problem for ecological analysis, right? Well, for this reason though, we made sure that our method provided as reliable estimate of uncertainty as possible. In this graph, you can see how the uncertainty score assigned to each prediction, what we call the confidence score on the x-axis, lines up pretty nicely with the actual rate of correct classification. This essentially means that for predictions with a high confidence, let's say 0.8, we can assign pretty much 80% of probability of being correctly classified. And this information is extremely useful for many reasons. First, we can use a threshold to disregard all those labels for all those trees that have a confidence score that is lower than an acceptable risk threshold. Let's say if you don't feel comfortable in using species classification for which there is less than 85% of probability of them of being correctly classified, you can throw them away. And by throwing them away, you can go down from the original 6 million trees per site on average to 4.5 million trees per site on average, which is still a fairly decently high number. Second, identify regions of higher uncertainty has also the potential to identify species and ecosystems that were not included in the original classifier. This gives us the opportunity to create a linkage between derived products and future field campaigns, which may allow us to optimize where in the landscape we should locate the next field plots to get the most interesting information for the future. Anyway, we use this pipeline then to generate surveys of species and size for about 53 million trees, for a total surface of about 400,000 hectares. I mean, roughly speaking, the full data set corresponds to 20 times the volume of the individual trees surveyed across the US every 5 years by the forest inventory and analysis, and all this for a fraction of the cost. Moreover, this pipeline has the potential to be applied across years, allowing us for monitoring whether and where we can observe changes in tree demography and properties. And if you ask me this, to our knowledge, it's probably the first attempt to produce a geo-explicit large-scale inventory for dozens of millions of individual trees spanning from Alaska to Puerto Rico. And this is even more exciting to us if we consider that soon we will integrate this data set with the estimates of leaf traits for all these sites. Together, the surveys, species, structural, and functional biodiversity, and can be used to unlock cross-scale investigations of ecological patterns and processes across and within a wide variety of landscapes, not only in the US, but eventually even beyond, wherever we have both remote sensing and high quality field survey. There is, however, a caveat. Despite how we managed to build ways to scale individual trees' properties for a continuum of scales, from patch to landscape, currently it's pretty much not possible to create surveys of billions for a continuum across tree to landscape to continent, or even so globally. In fact, the resolution of new multispectral and hyperspectral satellite data is still too coarse for distinguishing individual tree objects. However, many ecological questions require investigating tree properties for as good as a continuum of scales across wide regions as possible. For example, I've been talking before about trying to identify the role of intra-species as opposed to interspecies variability on traits distributions and trade-off, the leaf economic spectrum, right? Yeah, our derived survey can be useful to compare different landscapes, but definitely does not suffice to scale across regions. A solution in this case may be integrating complementary field surveys to enhance high density data set of tree species, like the forest inventory and analysis, with traits data collected only for a small number of locations, like in NEON. This way, it is possible to leverage the information from species and traits measures collected for a few hundred of individual trees, the black dots over here, and infer traits distribution for all those trees sampled across the forest inventory and analysis, the blue dots. And so this strategy is actually not even entirely new either. And in fact, the FIA has been used in the past to estimate traits distribution across the eastern United States. However, it has been done so mainly at the level of species, either directly or by building species distribution models. These approaches are generally referred to as community-weighted models, and they are based on this idea that large-scale variation of traits arise as the consequence of shifts in species, with traits mostly conserved within species, rather than being directly the traits affected by the environment. Therefore, these approaches assign species to their average trait value and completely ignore the intraspecific component of large-scale traits distribution. An alternative approach that allows for continuous estimate of traits relies on quantifying directly the relationship between the leaf traits and the environment, often represented as soil, climate, and/or topography. Despite this approach may contain some of the latent information about species, it failed in acknowledging those evolutionary constraints that reduce the realized range of traits value for particular taxa or for particular plant functional types. Therefore, despite both approaches made important contributions to our understanding of how traits scale geographically, alone, they have limited use in investigating the relative importance of intra and interspecific variation on the traits distribution and on leaf economic spectrum. And that's where we come into play. For this reason, in fact, we developed a joint hierarchical model that accounts for both the environmental and the evolutionary drivers directly on the leaf traits distribution. This approach not only allows us for better estimations, but also allows us for quantitatively assess the relative contribution of the intra and the interspecies source of variation. The environment here is consists on large-scale climate data and topography. Evolutionary drivers here consisted on species identity and the phylogenetic relationship among species derived from the open tree of life. And this is a key component because this is why we can make estimates for a bunch of species that we didn't have in the original traits dataset. To separate the intra and interspecific contribution, we used the variance partitioning. Intraspecies trait variation was considered as the variability in traits explained only by the environmental drivers, while the interspecies traits variation was the one that can be attributed solely to phylogenetic drivers. Whatever is left in the middle is essentially the variability that cannot be uniquely ascribed to either one, so neither to the environment or the species, because the two drivers essentially co-vary. Anyway, this way we could disentangle the two components. And our results showed that both of these components play a key and often complementary role in a traits distribution. In this slide, for example, you can see like the highlighted green boxes. They are the interspecies component of leaf traits distribution. And in general, that interspecific variation confirmed to be the dominant driver and explained around 29% of the total variance observed at a continental scale. However, the environmental driver, which is the orange boxes in here, had still a fundamental role in capturing what we think it is the intraspecies variability, which appeared to be the dominant driver for some traits like the pigments, and also accounted for a fairly decent amount of variation. We are talking about 13% on average of the total variance alone. But this is just looking at the data from NEON, right? So essentially, we're still looking from a very limited number of sites across the continent. But what is the role of the environment and within species distribution across a large geographic continuum that can be as wide as the species realized niche? Well, to answer this question, at least for the eastern of the United States, we used our joint traits model to make predictions on every individual tree sampled from the FIA, producing a data set of about 1.2 million trees. And this data can be used to explore geographic patterns of leaf traits and their economic spectrum for about 200 species. Now, our results suggested that for about half of the species, leaf traits distribution was significantly affected by some environmental drivers. But moreover, we showed that disregarding the direct effect of within species traits distribution will result in ignoring a gigantic source of variation that often corresponds to almost half of the total range for all species in the entire region. And this actually is what you can see here in the bar plot on the bottom, which represents the range of variation in nitrogen percent for 3 widely distributed species. The top 2 rows represent field observations from NEAR, and another source of field observation, which is essentially one of the biggest traits data set, it's called TRI. The 2 bottom rows represent predictions from our model, and on the top, with, and on the bottom without intra-species variability. And as you can see, how much variance is lost potentially by using a classical community-weighted modeling approach. Finally, our results suggested that the environment may be affecting traits distribution differently, depending on how far individual trees are from the edges of their species distribution. The bottom panel in this figure represents trends of abundance for different genuses as they shift across a latitudinal gradient. And the 2 panels on top represent the divergence of the environmental component of the model, the orange contour, and the evolutionary component of the model, the green contour, from the full model. Now, what this plot suggests is that as individual trees get closer to the lower edge of their distribution, the environment kind of like seems to, I don't know how to say in English, but essentially encourage a reduction in nitrogen. And this is possibly because when temperatures are higher than the species-specific optimal growth, leaves may downregulate their foliar proteins, and therefore, consequently, the amount of nitrogen to balance the photosynthesis and respiration. And this can have incredible impact on how we know and how we predict future shifts in species distributions. In fact, in conclusion, we believe that further investigation of the role of different ecological drivers on forest traits and species distribution is fundamental to better understand how we can predict future shifts in plant diversity and function. Moreover, as more traits, remote sensing, and survey data are being released for public use globally, leveraging these methods allow us to study dimensions of plant diversity across scales essentially that goes from the individual to global. But does it mean though that only computer geeks can access this information? Well, absolutely not. All the original data and the data products and the code of our work is and will be openly available for the community to download and to use directly for any of their ecological questions and applications. And this data is generally released as geospatial data, raster and polygons, but also as the more classical comma-separated surveys, essentially what the same Excel sheets that you would get if you go in the field and collect species and tree size and leaf traits. Moreover, we are also developing open software to make it possible for other folks to apply all those approaches on their own images and data set. And finally, we are also organizing data science competition to promote international collaboration and collectively develop, evaluate, and compare new approaches for the better of the entire community. And with this, I would like to acknowledge Morgan and Ethan, that are wonderful scientists and definitely even much better human beings, which is very, very hard to be because they're already amazing scientists. And I want to acknowledge my lab mates, my collaborators, my funding sources, my friends and family. and also all those crazy folks cheering from Italy and the whole continent. Bella.
Yay. Thank you so much, Sergio. That was an awesome presentation. Do you feel like a genius now after all that work? Or?
They're just too kind to me.
Well, we have a few minutes. So if you have any questions for Sergio, if you are in the Zoom meeting, you can unmute your mic and ask him or type in the chat. And if you are on YouTube, you can ask questions as well just by typing in the chat there. So please go ahead and ask questions now if you have them. Well, I'm not seeing any questions, but you are getting a lot of celebration in the YouTube comments. A lot of awesome work and great jobs.
Thanks, everybody.
Yeah, if we do not have any questions, then that will be it for our presentation today. And thank you so much, Sergio, for joining us and sharing your research with us. And for those in the class, we will see you next week. Thank you so much.
Everybody. See ya.